speaker of the session is David Kozlicki from Oregon State University. He's going to be talking about Quikr and Quikr, yes, Quikr, rapid bacterial community reconstruction. Too much wine for lunch via compressive sensing.
Thank you. Yes, and thank you to the organizers for giving me this opportunity to give this talk. So this is joint work with Simone Foucault, who's at UGA, and Gail Rosen, who's at Drexel University. So basically what I'm going to be presenting today is a bacterial community reconstruction tool. And I don't call it taxonomic binning because while it results in pretty much the same thing in your downstream analysis, the way it goes about it is a little bit different, and we're going to emphasize that as we go on. So here's the outline of our talk. Just state the problem really briefly, what people do now, and then take a little segue to talk about the mathematics. And by the way, if there are any questions about the math or pretty much at any point during the talk, please feel free to stop me. Then I'm going to talk about Quikr, and Quikr is for 16S rRNA, and then WGS Quikr, as you might guess, is for whole genome shotgun data sets. And this work has been published in Bioinformatics and PLOS One. You can get preprints at my website right here, and there's already software packages up on GitHub and SourceForge in hopefully one of these languages will suffice for your purposes. So let's go ahead and start at the beginning, talk about community reconstruction. So typically with this talk, I had a lot more slides going into detail, but I'm pretty confident that everybody now knows what bacterial community reconstruction is, but just a quick refresher, we take an environmental or health-related sample, get a bunch of short DNA sequences from it via whatever sequencing technology that you want, and at the end get a composition of bacteria, one of these pie charts, as talked about in the beginning. So this step right here is what I'm going to be concentrating on and what this tool addresses. How do you go from these short reads to this composition of bacteria? Okay, so in this workflow, I want to figure out what is this pie chart going to consist of, and then also what's the width of each one of these slices. So what's there and how much? And I'll go ahead and give away the punchline. What we did is came up with this tool, Quikr and WGS Quikr, that on simulated data at least is ridiculously quick. And depending on how you train it, can be very, very accurate as well. Okay, so current approaches. What do people do currently? Well, read by read is basically at the very highest level the kind of paradigm that people are using to be able to answer this reconstruction question. So as an example, if you have your data set and it looks like this, what they're doing is they're taking an individual read and then doing, well, a bunch of computations on it. And it can be different kinds of computations depending on what kind of tool you're using. So you could be doing string matching or pattern matching, like if you're using metaflan. Or you could be using Bayes' theorem, like if you're using a naive Bayesian classifier. Or you could be doing something BLAST-like, like with Thimble. But basically, at the end of the day, you're taking a read and you're doing some computation on it. And then from that computation, you get your classification that looks like this. And then you go to your next read, and you do the same computation, you get another classification, and so on and so on. Well, looking at this problem, I have a couple criticisms about this. What are my criticisms? Well, it's somewhat slow, because as sequencing technology progresses, we get more and more and more reads. So just by the technology progressing, our algorithms are going to automatically get even slower, because we have to go read by read. And in addition to that, typically we can't incorporate all the previously known information that we have. Why? Because if you're classifying against a database, then your algorithm's complexity is also governed by the size of that database you're comparing against to. So typically you have to use a very small, highly curated database, or some, very specially picked clade-specific marker genes and stuff like this. As an example, let's look at a ribosomal database project. So RDP has a database of about 2.6 million 16S rRNA sequences. But if you look at RDP's naive Bayesian classifier, that is RDP's NBC, I think training set 7 has only 10,000 sequences. So you're not incorporating, or you're incorporating a very small amount of sequences. This can have benefits depending on what your algorithm is doing, but basically we're incorporating little bits of not as much information, and it's rather slow. Okay, so that's basically what people are doing now. Stated the problem. Now let's take our segue into the math. Compressive sensing. What is compressive sensing? Well, you can make it really, really complicated and very difficult, but I'm going to simplify things as much as possible. So compressive sensing is basically a group of ideas that's at the intersection of mathematics, computer science, and statistics. And at the end of the day, it all boils down simply to linear algebra. So, if you recall linear algebra, we have this matrix equation, ax equals y. A is a matrix, x is a vector, y is a vector. Basically, we know a, we know y, and we just have to solve for x. Well, if we are in the case where this system of equations is underdetermined, we can have infinitely many solutions. I.e., so if our matrix looks like this, so it's this like short, fat matrix, then a priori, or in general, we can have infinitely many solutions, because our vector x is going to look like this. That's what we're trying to solve for. But the information that we have, y, is a lot less. Well, you don't have to completely give up at this point. If we make one further little assumption, we can actually make this intractable problem become tractable. How so? Well, if we assume that x has many zeros, that is, x is a sparse vector. And there are many cases where this is actually accurate, and we'll see later on that in our application it is a very reasonable assumption. But if x has a whole lot of zeros, then out of the infinitely many solutions that you have, you can pick the sparsest, or the vector that has as many zeros as possible. And then you can solve this equation ax equals y. So if you naively write down this sort of optimization routine, what it would look like is the following. So we are going to minimize over x the L0 norm. What is that? That's the number of non-zero entries in x, subject to satisfying this equation. Well, that seems all well, fine, and good, but unfortunately, oh yes, and geometrically, that's basically taking the hyperplane defined by these equations, trying to minimize the number of intersections with those axes. That's all well, fine, and good, but unfortunately, that's NP hard. There's no known polynomial time algorithm to be able to solve this. But, and here's the like the big insight of compressive sensing, is that you can replace this norm, which is causing the NP hard difficulty, with another norm. Let's just simply replace it with the L1 norm, the sum of the absolute values of the entries of this vector. Then, under certain situations, you can actually prove that the solution to the NP hard algorithm can be obtained via this convex optimization routine. And this is called basis pursuit. And the way they do this is basically we're just changing the geometry of the problem. And so then basically we're looking for hyperplanes intersecting this unit ball right here. In basis pursuit, there's another slight modification of this that is kind of like a regularized or penalized regression. And that's called basis pursuit denoising. And that's going to play a big role a little bit later on. So this works, but basis pursuit denoising, instead of requiring that ax equals y, I'm just penalizing with a factor. So ax has to be near to y. And that will help if the data that we're looking at is noisy. And as you might imagine, as lambda goes off to infinity, basis pursuit denoising gives you the same solution as basis pursuit. So that's compressed sensing in a 4-minute nutshell. Is that okay? Questions? None? Okay. Well, what does this have to do with metagenomics? Let's try and connect the two up now. So, as I said, we have this matrix equation, ax equals y. And how we want to think about this is A is going to be our database. That's the thing we're going to be classifying against. That's all our known information. X, that's going to represent the concentration of bacteria. So each one of the entries of X corresponds to a certain bacteria, or maybe a certain phyla or taxa or OTU. And that entry of X is going to tell you what concentration that certain bacteria or organism is in your sample. And then Y is going to be our actual sample. Well, how do we actually get this to make mathematical sense? Well, to be able to do this, we have to take a Kmer-based approach. And the Kmers that I'm talking about are really rather small. So only like 5, 6, or 7 is the Kmer size that we're talking about. Now, how do we form this matrix A? Well, given a database sequence, we're going to calculate the frequency of all the Kmers in it. So we take a known bacterial one, like go to RDP's training set and take one of those sequences and calculate all its Kmer frequencies. Add that to your matrix A. Then go on to your next bacteria, and then your next one, and then your next database sequence, etc. And you're going to form up this matrix where the columns are indexed by sequences and the rows are indexed by your Kmers. And note that the sum of each one of these columns is going to equal 1 because we're talking about Kmer frequencies. Yes, it's just what I said. Okay, so now we know how to form A. What about Y? Well, given a sample, we're going to calculate the frequency of all the Kmers. So this is basically the compression step of compressed sensing. We're taking this massive sample and compressing it down to just be a vector of size 4 to the k, which is really rather small and actually rather easy to store and send to other people, etc. So it's an extremely large compression step. If you do this, so y is your frequency of Kmers in your sample, then ax equals y under moderately acceptable suppositions. Okay, so we've been able to fit the metagenomic problem that we're trying to do, that is estimate concentration of bacteria, into the compressive sensing framework. Okay, that's great theoretically, but how does it actually work in practice? Well, remember this basis pursuit denoising that I was talking about? It works, but it's kind of rather slow. And basis pursuit denoising is actually not taking into consideration all the assumptions that we have in this problem, because if you recall, we were trying to reconstruct this vector that's non-negative, right? It's supposed to represent frequency of bacteria. Well, if we do the following, if we simply replace this L1 norm right here with the L1 norm squared, which might actually be a little bit more natural because notice we have a square on this norm too, then it turns out that using this assumption that the vector we're trying to reconstruct is non-negative and that the column sums of our matrix are bounded by a constant, in this case 1, we can reduce this thing to least squares. Okay, what do I mean by this little matrix guy right here? So this is a matrix, and this means that the first row is 1s, and then the rest of the matrix is root lambda times A. So that's not a division sign. That's just telling you different rows. Same thing over here. This is a column vector where the very first entry is 0, and then y is everything down here. Now this is actually really rather surprising, and one of these cool examples I think of where the math informs the biology and the biology informs the math, because the reason why people were looking at sparsity promoting techniques in the first place was because least squares overfits terribly, right? But in this case, just modifying it ever so slightly, these two are exactly equivalent. So actually there's a short note that Simone and I have in IEEE transactions on signal processing that details when this reduction is, when it holds. So when you get unique reconstruction. Please do.
I mean, under the X to be positive condition, this is equivalent to L1. What is it? X is, all the elements are positive, right?
Correct.
So the L1 norm is the sum of X.
Precisely.
Then you take a square of that sum?
Precisely.
And is it equivalent to the previous formulation you said, or
Yes.
not equivalent? Equivalent.
It's equivalent. Yeah, because if you take this guy and you simply, do out this multiplication, basically we're going to have the sum of the entries of X minus 0 squared. Okay, well, the sum squared, and if you take an L2 norm of just a scalar, it's just the scalar itself. It'll be this guy.
Yeah, under the X being positive.
Correct. Yes, it has to be positive. So this is non-negative least squares. Yes, I guess I should say non-negative right here. Non-negative least squares. Question?
Why the matrix A is in the denominator. What does that mean?
So it's not actually a denominator. I'm just trying to say that this is emphasize that this is a first row. So it's a matrix, and the first row is ones, and then below that is
Oh,
A.
that what it is?
Yes,
Okay.
yes. Okay. So basically, let's summarize. Quikr algorithm, what is it? It is using a K-mer-based approach to reconstruct your vector X. Notice I'm not actually assigning individual sequences. I'm assigning an entire sample, basically. So it's not quite taxonomic binning. But you train it using whatever database of sequences you want. You calculate K-mer frequencies in your sample. Use non-negative least squares, which is ridiculously quick. And since it's a quadratic iterative K-mer-based reconstruction technique, I call it Quikr. Okay, well, let's see if this is any good in actual practice. So let's talk about Quikr. Now, Quikr, that was the first one we developed. That's for 16S rRNA. So I'm going to talk about 16S first, and then I'm going to go on to whole genome. So let's see here. As I said, it's extremely quick. So I'm going to compare it to RDP's NBC. It's just the easiest thing to do. So while RDP's NBC is linear in the number of reads in complexity, turns out Quikr is really rather fast. So if you take a log plot of the number of reads in your sample versus the number of seconds, turns out that we can classify classify a million reads or classify a million read sample in well under a minute. It also has a really small memory footprint, so you can actually do it on a laptop if you want to, depending on how you train it. But it's not going to be any good if it's very quick and it doesn't give you at least a somewhat accurate reconstruction. So let's go ahead and see how it works on simulated data. So I used Grinder, which is this really nice 16S as well as whole genome shotgun read simulator. And I just simulated a whole bunch of different data sets, a whole bunch of different reads, and a variety of different parameters. Let's see how this works. Okay, well, we have a choice when we form this equation, ax equals y, of how we choose A, right? Because we have a choice about what sequences we're going to classify against. So I'm going to do two different cases for 16S. So in the very first case, I'm going to use this K-mer training matrix to be the same as RDP's NBC training set 7. So that's the 10,046 highly curated selected sequences. If you do that, here's our timing. Well, in comparison, well, if you look at our median, we do 25 seconds, RDP's NBC 6 hours. But as I said, you could train it a little bit differently. Let's try to incorporate more of the previously known information into this. So what I did is I took the entire RDP database and then I ran it through tax collector to be able to get the well-labeled sequences. And we ended up with something like 275,000 sequences. And I'm going to make that be my sensing matrix or my K-mer training matrix. So small matrix, large matrix. So in this case, as you might imagine, since now this matrix, instead of being 10,000 by 4 to the 6, it's now 275,000 by 4 to the 6, we get an increase in execution time, which we do, but it's still woefully faster than RDP's NBC. Question? Yeah, so the K-mer, it's actually included because we're only talking about K-mer size like 5 or 6, which is really quick to use. Any of the well-known K-mer counting techniques, jellyfish or whatever. We actually just use a custom-made C program, and it takes literally just a few seconds on a 800,000 read data set. Another question?
So are you talking about training or about prediction here? So you mean what is fast? Prediction is
Prediction,
fast,
yes.
but prediction is never bottleneck, right? I mean, if you look at any kind of a
It's certainly a bottleneck in this case because the prediction that RDP's NBC takes hours.
okay. Well, prediction with standard least squares is
I'm talking about predicting the concentration of bacteria in the sample, not predicting a least square solution. Right. And what about the accuracy of this prediction of bacterial concentration? So, there are a bunch of different ways you can use unifrac or different distances and stuff like that. I'm going to use one that's a little bit closer to just the compressive sensing literature, the L1 norm. So basically, if you have predicted bacterial frequencies and actual bacterial frequencies, the error that I'm going to be talking about is L1. So you can think about like the total error, basically.
Your X is not human frequency, right? You renormalizing the frequency.
Yes. Everything's normalized to sum to 1. Right. And if we do that, if we use that really small 10,000 sequence database to train our method that has formed this matrix A, then here's what the plot looks like. Blue is Quikr, red is RDPs in BC, and we have less error at higher taxonomic ranks. We have the same exact error, which is kind of surprising, at the family level, and then RDPs in BC has less error at the genus level. But the real power of this method that I was saying is that you can incorporate more information. So let's see what happens when you move to that larger database. If you move to that larger database, then at every taxonomic level, we have much lower error than RDPs in BC. And if you tried to use a Bayesian classifier using those 275,000 sequences, I think it calculated something like 5 years to be able to make, to reconstruct a single sample. So, you can talk about other error metrics as well. I'll go through this just a little bit quickly because I want to talk about a whole genome. But basically, if we have precision, sensitivity, specificity, and accuracy, so forget about concentrations, just talk about presence or absence of the bacteria. Then once again, training with the smaller matrix, we have pretty bad sensitivity. But when you move on to the bigger matrix, then pretty much all the different measures become somewhat comparable to RDPs and BC if you're just talking about a binary classification task. Okay, well, when would you actually want to use this tool? So you want to use this tool basically when you want to do some sort of a little bit higher level look into many, many different samples. So as an example, on real data, we took 5,000 samples from the Human Microbiome Project, which is about like 2.7 gigabytes worth of data. And we were able to classify all 5,000 of those samples in less than 8 hours. And doing this with RDPs and BC would take like 12 days or so. And if you want to do one of these fun like PCOA plots to see how these things cluster, well, this thankfully was a previously studied data set, and you get a clustering that looks like this. If you do the same thing with Quikr, then you get at least qualitatively similar clustering. So, here gastrointestinal is off from everything else, and the skin and urogenital kind of crossover, etc. And it can do this, by the way, on a desktop computer. So RDPs and BC, this is running on a cluster. Quikr, this is just running on your desktop. So if you just want like a real quick look into your sample and you have tons of different samples, you can use Quikr to be able to generate these PCOA plots very quickly. Okay, oh yeah, another thing is you don't have to do chimera filtering because the way this counts the k-mers, so chimeras we know are an unavoidable artifact of 16S RNA, amplicon sequencing, or basically we get sequences that are half E. coli or half staph, But it turns out that the k-mer count of these sequences are basically k-mer count of this sequence plus k-mer count of that sequence. So they don't really hurt our method at all. In fact, so if you simulate these, check out this plot. Here's percentage of chimeras in a sample, and here's our error at the genus level. So even if you go up to like 100% chimeras, the method is still not negatively impacted, which I doubt you can say with other methods. And this is only because of this fun little k-mer counting fact right here. Okay. Did I have half an hour or 45 minutes? Half an hour, okay. So in the last 3 minutes, let's talk about WGS Quikr. Okay, so whole genome shotgun, because everything's moving to whole genome shotgun. This is the technologies that we want to be using. It's almost the same exact setup as a 16S. So in this case, our k-mer training matrix is going to be using whole genomes. So I went and there's something like, 2,500 whole genomes in NCBI's RefSeq whole bacterial genomes, and like half of those are just strains of other ones that are in there too. So I picked about 1,400 of those and formed these, the matrix or the columns of this matrix. So basically you have an entire whole genome and you count the k-mer frequencies in this guy and set that as one of your columns. And you do that for all 1,400 of these guys. You're still doing the AX equals Y. Everything still stays the same. But here's the important difference. Because 16S rRNA is relatively short, it's not an unreasonable assumption to say that the k-mer frequency in the first half roughly reflects the k-mer frequency in the second half. That same assumption doesn't hold for whole genome shotgun, i.e. we have a whole lot more error in the measurements and in our training matrix. because we have different error, this regularization parameter is going to play a very important role. So when we're dealing with 16S, as you change this regularization parameter, basically the error just drops and stays flat. However, when you go onto the whole genome, what happens is as you increase this regularization parameter, the error drops. And then after this point, basically you start fitting noise because you're enforcing AX equals Y a little bit too much. So we need to find out, where is this minimized? So this regularization parameter choice, basically. Well, we do this adaptively. And the very cool thing in this case is if you look at the number of iterates at a certain lambda required to solve the non-negative least squares equivalent of this guy right here, you get this plot. So if you use a small regularization parameters, not many iterates. If you use a large one, many iterates. And the interesting observation is in every single one of the cases that we looked at, the lowest error occurred exactly at the highest rate of change. So if we take the first derivative or first difference of this guy, where we experience the highest rate of change is where the lowest error is. So basically we choose our regularization parameter by stepping along at different values here, taking first differences, and just finding where that's maximized. Or where smoothing spline interpolation of that is maximized. So it's a really good thing that this algorithm is very quick, because notice we have to be running it over and over and over and over again for all these different lambda values. So WGS Quikr is going to be a little bit slower than the 16S rRNA. Okay, so real quick results comparing to metafilar, comparing to metaflan. Once again, we have WGS Quikr having the pretty much flat execution time, compare number of reads versus number of seconds. And on simulated data, did a whole lot, like a billion reads of different WGS simulated data sets. The execution time, as I said, it's a little bit slower. Now we're talking about minutes instead of seconds. But it's still a little bit quicker than metaflan. But what about accuracy? Well, let's look at the reconstruction error. So here's a histogram of all those 720 simulated data sets. So this is the error right here. So the farther you go this way, the better the reconstruction. The more you go that way, the worse the reconstruction. And if you notice, Quikr, which is this purplish bluish one, has lower error on average than metafilar and metaflan right over here. Well, if you're worried about what kinds of simulation parameters affect your reconstruction, then I don't think I have time to go through these in detail because I have about 2 minutes left. Basically, more reads is better. Longer reads are better up to a certain point. Quikr can deal with extremely short reads like 35 base pairs where metafilar and metaflan can't. The more species that are present, the worse every method gets, and abundance models kind of don't matter except for exponential is the best for this algorithm. So just a real quick to guide your intuition about why this is working so well. Basically what Quikr is doing is very accurately reconstructing the most abundant organisms that are present in your sample. So here's an HMP, this is the even mock community. And if you take the actual frequencies based on gDNA mass, then Quikr gets a very high fidelity estimation of the most abundant organisms, more or higher fidelity than metafilar and metaflan, at the cost of not being able to reconstruct your very rare ones as much. So again, as I said, a very large, a larger level look into your sample with very accurate estimation of abundant organisms. Unfortunately, I didn't have time to talk about this generalization that we recently made, we being a group over in Finland. But basically, it's a new paradigm in community reconstruction because we can do the whole sample instead of read by read, and it can either be quick or very accurate depending on how you train it. So thank you for your attention.
That you were just showing. My question is a bit, I guess, broader than this example, but what I'm wondering is whether the metrics we are using to measure error are appropriate, because it seems to me that getting the highest relative abundance is what's driving the error metric more than the low abundance one. But in many cases, I imagine getting the high abundance is easy. You could use whatever method and you'll get them, right? But what you are more interested in many cases, not always, is getting the low abundance.
Right, so I would answer that with a couple things. So if we were using the L2 norm to measure that, then yes, that would be weighting the higher abundance ones as more important. But in this case, since we're using L1, we're just saying raw differences and sum up all those. And the other thing is, so you said you could use any method, but notice Metaphylar and Metaflan don't, they aren't able to. But, so you are correct in saying that there are a bunch of different metrics that you could use, and depending on which one, each method could be said to be better than the other one or not.
I guess my point was, if you use Metaplan here, you get 40% instead of 50, and for a lot of problems that might not really matter a lot if you think about proportions. But if you get something that's 2%, and instead you think it's 0.1%, that might matter, right?
Yeah, it definitely.
But
it just depends on what the biology question is that you want to ask. Absolutely.
In a slightly different direction. So my criticism of Metaflan, and this is unfair because I'm beating you with them, the whole classification happens on the DNA level, right?
Mm-hmm.
So we know that the space that we have represented in the database is a lot smaller than what we actually encounter. So you use a very small fraction of your metagenome when you're actually using Metaflan, and only a very small fraction is actually characterized, and you're extrapolating from that.
Right.
So what's the fraction of a real-world sample, not this mock community? Because this is a clonal sample that was put together and it has no strain variation, yada yada. Plus, we know all the strains.
Mm-hmm.
So, what's the fraction of a real-world metagenome, say from HMV and maybe from something that doesn't have this massive amount of genome sequencing backing it up? Because I'm afraid it might be very, very small, actually.
So you're asking what's the fraction of classified reads, basically?
So yeah, what's the fraction?
Yeah. So since we're classifying the entire sample at once, we can't actually say what percentage of reads, but you can look at your residual. So if your size of your residual is really large, that means you're not, there's many unknown species in there. If we went back to that PCOA bunch of plots, there's some real data, some nice messy real data.
But you wouldn't have an intuition as to how much you're actually.
No, yeah, you unfortunately, because of the method, you can't answer that question. Yeah.
Also about this, do you have any intuition about the error rate at higher aggregation levels? My first thought is that as you move up in the taxonomic hierarchy,
Mm-hmm.
and this classification is more dangerous. So
Yes.
And this all at the species level, these error rates that you're computing. So what happens up when you go to the family genus?
Yes, so as you might imagine, as you go to finer levels, your error does increase. And in experiments that I've done, genus level is pretty much where you'd want to stop if you want to be fairly confident.
Okay, just one more question, but then we have to move on.
Is it true that when you use one norm, then you have disparity, and then use x very small value of x? You try to solve by lasso or something like that, do you?
Yeah, so I'm using basis pursuit denoising instead of lasso. Yeah.
But using one norm and the square norm will make a difference, right?
Yes, but
one norm you got a disparity, many zeros, right?
right, but we can prove in this situation, because we're reconstructing non-negative vectors and we have that requirement on our matrix, that those two are actually equivalent.
We talk about
Yeah, we can talk.
Okay, thank you.
Thanks.